Hello everyone, my name is Kalen Rasmussen and I am excited to present to you a portion of my research concerning silicified hydrothermal microbial communities. To begin, silicification is the saturation of organic matter by silica that may result in the formation of resilient microfossils. Also, silica colloids are very small and are fantastic at preserving minute features of microorganisms. This is important for two main reasons. In industry, silica scaling is a major issue in geothermal power plants, and microbes have been implicated in this process. Also, within the rock record and ancient earth, silicified organisms may act as a window to the past, where researchers may use their presence to further understand ancient environments and life. A great place to study silicification is Yellowstone National Park, which, due to the Yellowstone hotspot, has a plethora of silicifying hot springs. One such hot spring group is the Sentinel Meadow group, shown here. Each white spot on the satellite image represents a silicifying hot spring. Here, water supersaturated with silica rise to the surface where the water is cool and silica precipitates. For this study, we chose to work at Steep Cone Geyser, circled here in red, which is a slightly alkaline surface boiling thermal feature. Steep Cone is a unique feature in the park. First, as one can see in the upper right, Steep Cone rises above the meadow approximately 5 meters. Next, there are living yet actively silicifying microbial mats on the top and the outflow channels. Lastly, Steep Cone possesses a rockfall, which exposes layers of fully silicified samples, allowing one to project back in time. It is thought that hot springs are a consistent environment, so examining microbes and connecting the living microbial community to the rock record is possible. For the study, a combined approach was used to examine the biology and geochemistry of the silicifying environment. We collected samples at 1 meter increments along an outflow channel over time to track the geothermal and biological changes occurring. Upon examining the site with the eye, one can see an evident visual change. It changes across with time and down the outflow transect. To determine how this change manifested in the microbial community, DNA was extracted from the biological samples and the 16S rRNA gene was sequenced to better understand the microbial communities. These data were used to construct an ordination using a weighted unifrac distance matrix to examine how the samples related to one another and changed. Here, one can see the results of the distance matrix, which allows us to see shifts in the beta diversity of the microbial communities. First, examining samples down the outflow channel, one can see a general trend to the right and up for the two of the sampling dates. However, examining two other dates results in a different and varying trend. These results make it difficult to determine general overall trend with outflow. Next, if we examine the shifts of the microbial communities to distinct locations with increasing time, we can see clearer trends. For the first to second time voids, a span of almost 8 years, there is a general shift to the left. Next, there is an overall shift down for samples taken in the spring, summer, and fall of 2018. Lastly, there is an overall shift to the right, while also bidirectionally from 2018 to 2019. These results suggest that while there are not distinct diversity trends along the outflow channel that apply to every sampling time, there may be distinct trends associated with the passage of time, with larger shifts occurring over larger durations. A few parameters that may dictate microbial trends are pH and temperature. Here one can see steep gradients for all sampling times. Temperature has a decrease in almost 50 C down the transect, while depending on the sampling time, the pH may shift as much as half a unit. Now let's dive into the most abundant microbial taxa inhabiting this environment. Here we have a heat map showing the top 15 most abundant microbial taxa using the relative abundance of 16S rRNA genes. The names are listed as class then genus. We do see that overall the inhabitants of the steep cone outflow transect are typical of geothermal environments. Next, if we dive into potential associated metabolisms of the abundant taxa as inferred by 16S rRNA gene sequencing, we can see that the system possesses a large number of potential phototrophic capable taxa. Additionally, there are a large number of taxa associated as chemotrophs. Then, if we delve more into the potential metabolisms, we can see that a number of microbial taxa are closely related to microbes capable of sulfur cycling. Next, we can examine the shifts in relative abundance of microbial taxa with time. Here, one can see that while one taxa may decrease, another microbe may increase to fill potential niches within the microbial community. This is especially linked to corresponding shifts in potential phototrophs and possible sulfur cycling organisms. Examples of these shifts are circled here. Here, Rosiflexus may shift to Geitleridema, both phototrophs, and Chlorobia may shift to Tepidomonas, both lithotrophic capable microorganisms. Examining trends seasonally, again we see a shift in abundance that may be associated with potential sulfur lithotrophy. Moving forward in time again, we see two more shifts in microbial abundances that may correspond to the dominant phototrophs and or sulfur cyclers changing along the transect outflow. Also of note, with time we can see an overall trend that shows a decrease in certain taxa. Lastly, we can examine potential morphology as inferred by 16S rRNA gene sequencing. We see a range of morphologies from coccoidal and tetrad to filamentous, which seem to represent a large portion of the microbial community's abundance. Also present are rod-forming taxa, which under different environmental pressures will form filaments. Lastly, individual rod-shaped morphologies such as Chloroacidiobacterium are present as inferred by 16S. Next, we can examine silicifying trends along the outflow using SEM imaging. At the source of steep cone, we can see two textures, one, smooth and finely cemented texture, and two, a popcorn-like texture made up of aggregated silica colloids. However, we see there are filaments that are preserved within the source samples at steep cone. Moving down the transect to 1 meter, we again see two textures, smooth and aggregated colloids. The aggregated colloids and well-cemented smooth fabrics are layered with depth, as displayed by the bottom images. Additionally, in the upper left, we can see that the smooth layer is able to preserve casts of microbial cells. A silicifying microbial biofilm displayed on the left is also present at this location. Here, one can see the living microbial biofilm, composed of randomly oriented microbial cells, is dominated by filaments and potentially connected by EPS. The layer immediately under the biofilm, shown on the right, is also exposed, revealing a highly silicified microbial cell matrix with silica colloids attached to the cells, and also with some infilling by colloids. As we move to the last sampling point down the transect, we see some similarities and some differences. On the left, we again see the gooey microbial biofilm, which appears thin as portions of the underlayer, which is completely silicified, are poking out or exposed. Additionally, in the upper right, one can see that the silicified underlayer contains completely encrusted microbial cells, as well as higher degree of infilling and cementation due to silica precipitation. Furthermore, we are able to see the ability of silicification to preserve fine details of microorganisms. Here, both the sheath and cell wall have been well preserved by silicification. On the right, the sheath silicified area around the cell wall is highly cemented. Next, we can examine the rough potential magnitude these microbial taxa play in these silicifying environments. Here, we can examine the relationship between the abundance of taxa versus the transcriptional activity by comparing the relative abundance of the 16S rRNA gene, the y-axis, versus the relative abundance of 16S rRNA in the x-axis of select abundant taxa. The dotted line represents a microbe that's transcriptionally active and is equal to its represented abundance within the microbial community. Thus, a microbe occurring above the line, such as Meothermus, may be overrepresented in the DNA fraction with respect to its potential metabolic impact, while microbes occurring below the line are underrepresented with respect to their potential microbial activity. Thus, the activity of abundant taxa, such as Leptococcus and Tepidomonas, may be underestimated. We can also zoom in and view samples that were less abundant. As one can see, this trend also applies to corresponding taxa at lower abundances. In conclusion, these data show that steep cone is not a static system. The microbial communities that shift more over longer time spans, and the outflow ecosystem may support a range of metabolisms. Also, steep cone hosts the active microbial community that is continuously silicifying and sits atop an infilling, fully silicified microbial matrix. Lastly, this work shows that by solely examining the rock record, one may underestimate the ancient, diverse microbial communities and their activities that were present. Lastly, I would like to thank all those that have helped me perform this research and the organizations that have made it possible.